Go ahead and get started again. Can everyone hear me all right? Okay, great. So my name is Matt Spitzer. I'm a graduate student in the Nolan Lab here at Stanford, and I'm going to talk a little bit this morning about reagent development. I'll focus the first half of this talk on antibody conjugation for the mass cytometry workflow. So it's a little specific to CyTOF, although the chemistry is fairly similar for maleimide-based fluorophores as well. And then the second half is really going to be on validating reagents. So that applies equally to mass cytometry and fluorescence-based flow, whichever you may be more interested in, or both. So a brief outline. I'll talk, as I mentioned, first about antibody conjugation, a bit of overview on the chemistry that's involved in that, and then I'll walk through a simplified version of our conjugation protocol to give you a sense of kind of how much time it takes, how much effort. I'll walk through some titration data, how to optimize the reagents that you've generated. And as I mentioned, that really applies to fluorescence-based flow as well. And then some troubleshooting tips if things aren't looking quite as beautiful as you would hope they might. And then I'll touch a little bit on a couple other tools that we've developed, a viability stain that we use for a lot of our samples now, similar to like Aqua or PI that you might use on fluorescence, and then a couple other reagents that we use on mass cytometry. So as Sean mentioned, how do you get over 50 parameters? This is theoretical. We're not quite there yet. It's by really taking advantage of the variety of stable isotopes that exist even within elements of the lanthanide series. So as he mentioned, europium is a great example. You see the average atomic weight is 152 AMU. But of course, that really comes from a split of 151 and 153. And if we isotopically enrich those, we can measure them both very well on the mass cytometer. And so going back to a slide that Sean showed, every vertical bar here is a stable elemental isotope that is measurable in the mass range that we record. And to give you an idea of how we utilize those on the mass cytometry platform, you'll see the antibodies are primarily labeled with metals of the lanthanide series. We use a DNA intercalator that's iridium-based, although there is another one that's rhodium-based right above it, so it has very similar chemistry. And then we now use these palladium isotopes to barcode our reagents, and that's something you'll hear a bit more about tomorrow. But essentially, it's a way for us to mix divergent samples, stain them all in one tube to remove technical variability, run them on the machine at the same time, and then deconvolute the data back out into the samples that you mix together. So a bit of background on the chemistry that we use to label our antibodies. We chelate heavy metal ions, trivalent ions, using DTPA, diethylene triamine pentacetic acid. And you guys have all the slides in front of you, so feel free to take notes or follow along as you'd like. And that interaction between a trivalent lanthanide ion and DTPA is very strong. It's an order of magnitude stronger than avidin and biotin. So those ions aren't going anywhere once you've chelated them, which is very nice for us. We can make master mixes of lots of chelated antibodies and leave them in the fridge for quite a long time, not needing to worry about ion switching. And using this sort of solution-based labeling strategy, we can typically label antibodies with between 100 and 300 metal ions. So getting a fair amount of metal on an antibody. And as you can see, these chelating groups are attached by a long polymer chain, which we then bind to the antibody. And the chemistry we use to do that is a bismaleimide type reaction, where we take advantage of the fact that antibodies have disulfide bonds, and we can dissociate those into sulfhydryls and react that group with maleimide, the structure you see on the top right. So we use a bismaleimide linker on a polymer chain that has a number of different chelating groups, each binding lanthanide ions. So as I mentioned, this technique does require partial reduction of the antibody to expose those sulfhydryls in the Fc portion. And the reaction really does approach completion. So we can label antibodies fairly standardly. Repeating the same protocol on the same clone of antibody generally gives very similar results, which is great for reproducibility. So how do you choose the right antibody to label, or where do you start in terms of developing new reagents? And this is maybe a little less critical now that DVS, the company that manufactures the mass cytometer, actually does sell some commercially available reagents now. I made this talk before that happened. So choosing a clone might not be such a challenge if you're using commercially available reagents. But if you take a look at that catalog and you have the biology that you're interested in mind, I would speculate that you would find that there are markers you want to measure that aren't on that list. So if you do want to develop a reagent of your own, where do you start? My suggestion would be to start with an antibody that's been validated for use with conventional fluorescence flow cytometry. You can look at the BD catalog, the BioLegend catalog, eBioscience, any of the big name antibody manufacturers. Generally a good place to start. Those clones tend to be fairly resilient and will stand up pretty well to the CyTOF or mass cytometry labeling protocol. I'd say from my experience, like 95 to 99% of those antibodies work pretty well when you try to use them by CyTOF. So provides a really nice means of comparison. You can run a sample on fluorescence, run a sample on mass, make sure that your same cells are staining with that reagent, and it's generally fairly straightforward. On the other hand, if you're interested in a protein to which there's not a great commercially available fluorescently labeled antibody, it's still very possible to develop CyTOF labeled reagents. It does require a purified, I say here monoclonal antibody, it's preferable, but you can also use purified polys. It does need to be purified. I'll mention why in a little bit. And the validation for that is obviously a little trickier. And it really requires having a good positive and negative control population of cells, as you can imagine. Sometimes that's a little bit trickier depending on exactly what protein you're trying to measure. So what do you need to start labeling antibodies for the CyTOF? Well, first, your antibody, generally a good place to start. So you need 100 micrograms of carrier-free IgG. And it is fairly important that it's carrier-free, especially BSA-free. And that's to ensure that the only sulfhydryl groups available for binding of the polymer are present on your antibody and not in your carrier protein. There are a couple carrier proteins that are compatible. Like glycerol, for instance, doesn't have any sulfhydryl groups, which is convenient. Or not glycerol, I'm sorry, gelatin. Glycerol would not be a protein. Gelatin doesn't have any sulfhydryl groups, so you can, but it's more difficult to determine your recovery and titrate the antibody because we use like a nanodrop, a spectrophotometer, to read out how much antibody we recover from the process. If you have carrier protein, it's a little trickier. It's better to be carrier-free. And then the polymer is commercially available from DVS Sciences under the trade name MaxPar. And that is that bismuleimide functionalized polymer that I showed you previously. A lanthanide solution, these come in the form of trivalent metal salts. Again, those are now commercially available from DVS. And we like to keep our stocks at about 50 micromolar in one of the DVS buffers, the loading buffers, just at 4 degrees. And then you'll also need a series of buffers. And again, these are available from DVS, a loading buffer, a reducing buffer, a coupling buffer, and a washing buffer. And I'll walk you through when to use each of those in a protocol. Also, a reducing agent, we use tris-carboxyethylphosphine, or TSEP, which is a fairly mild reducing agent. And that's to free up the sulfhydryls from the disulfide bonds in the Fc region of the antibody. And then an antibody stabilization solution to store your reagents when you're done with them. It's nice to keep them in a little bit of carrier protein. These are either TBS or PBS based, and we supplement them with a little bit of sodium azide for preservation purposes. And then in addition to the different solvents, we also need millipore centricon columns. These are just size exclusion columns that we use to swap out buffers. during the protocol, some screw top epis are nice to store your antibodies in rather than just the clippy ones, but of course you can do that too. And one of the really important things here is to use filter pipette tips for all the steps of the protocol to really prevent any metal cross-contamination. Even a little bit can throw you for a loop. So filter tips all the way. So a couple notes and guidelines on the general protocol. First, the concentrations are fairly important, and sticking to the order of the protocol is important because high concentrations of lanthanide metals will precipitate proteins. So as you can imagine, it would be more convenient for all of us if we could just throw some polymer onto an antibody and then throw a little bit of metal on at the end so we could switch up our panels more easily. But that's not really practical because adding heavy metal directly to your antibody will cause it to precipitate. Purification, it is possible to obviously purify antibodies if they only come in the presence of a carrier protein through a variety of methods that we can talk about if anyone has specific applications that they need to do that for. Melon cleanup is a really common one. And the timing throughout is fairly important. So I'll point out some of the steps that are pretty critical. So just a brief overview of how do we label antibodies. First step is, as I mentioned, to incubate our polymer with our solution of heavy metal ions. And preloading in this way prevents that antibody precipitation. Second step is to partially reduce our antibody using TSEP to expose that sulfhydryl group. And then incubating these together, we will allow the polymer to bind that sulfhydryl and form our conjugated antibody. So, brief overview. Again, the first step, adding our polymer to a solution of heavy metal ions. This takes about an hour to achieve. And about halfway through that process, you'll want to start reducing your antibody in a pretty low concentration of tris-carboxyethylphosphine. The reason we use a low concentration, for those of you who don't think about B cells or antibodies all that frequently, is that there are actually a lot of important structural disulfide bonds in an antibody. Now, very conveniently for us, the least important of these for antigen specificity lie in the Fc region, and they're also the most easily reduced. So the concentration of our reducing agent that we use is fairly important to ensure that the specificity of reduction is really restricted to those in the Fc region. If we use too much, antibodies can break, so we can actually separate the two arms of an antibody by fully reducing it. That's not a huge deal because you actually will retain antigen specificity, which is really directed through the FAB regions. But some antibody clones that require disulfide bonds in those regions, especially the CDR regions, can be a little problematic in terms of labeling through this protocol. And then the last step is to couple our partially reduced antibody with that metal-labeled polymer, again, producing our conjugated antibody, which is then ready to be used for the remainder of the mass cytometry workflow. So just a brief review. 3 main steps here. The whole protocol takes about 4 hours to complete, so it's not a tremendous time suck, and you can multiplex it. So when I label reagents, I usually do 8 or 10 at a time. It takes 4 hours, so you can label a full panel of 40 antibodies in 2 days if you really want to. So the aftermath. What do you do when you have a newly conjugated antibody? First is to quantify it, which is nice to ensure that you're using a reliable and precise concentration in all of your staining. So we just measure the absorbance at 280 nanometers on a nanodrop or some other spectrophotometer. Pretty straightforward. I'm sure most of you have used nanodrops before, or specs. And then we generally like to titrate our antibodies over a 6-step titration scheme from 8 to 1/4 micrograms per mil, which is kind of a nice dynamic range to test out just in 2-fold steps. And we usually stain in a 100 microliter staining reaction, which is pretty typical for flow cytometry in general, unless you're doing a ton of cells, and obviously you can scale that up. But what's fairly important is for the titration to have really good biological positive and negative control cell populations. So if you're using primary cells, That's often fairly straightforward since you'll have heterogeneous cell populations in your samples. If you're using cell lines, then it's great to identify cell lines that will or will not contain your antigen of interest, and to make sure that you run your titration on both of those to really get clean results. And then, as I mentioned, storage of the antibody, we like to use that antibody stabilization buffer that's commercially available from Candor. So now I'll walk you through a little bit of titration data and just sort of point out some of the things to look for. And as I mentioned, this really applies to fluorescence-based flow cytometry also. So titrating antibodies, as Peter mentioned, and I'm going to repeat because it's really important for phosphoflow, is really critical. A lot of things change when you start staining fixed perm cells rather than live cells. So really, really strongly recommend doing that before throwing important samples at the machine. So here's just a quick example of something fairly straightforward to titrate. For those of you who work in mouse, you probably know that there are these really convenient congenic markers that we often use for adoptive transfer experiments or making bone marrow chimeras, things like this. And one of them is CD45.1 and CD45.2. They're just two different isoforms of the phosphatase CD45 that's expressed on pretty much all hematopoietic cells. So here in this little experiment, I just tossed in half and half 45.1, 45.2 splenocytes from two different mice and stained them with an anti-CD45.2 antibody that I labeled. And as you can see on the far left, looking at the pooled singlets, we see a really nice, pretty much 50-50 mix of these two. And then when we start to look at our gated cells, just gating on 45 positive and 45.1 positive and 45.1 negative, you see that the 45.2 staining is really restricted to the 45.1 negative population, pretty much what you would like to see. And what I'll point out here is, depending on what's critical to you, if you're not really so interested in the biology of the phosphatase, you more just want to gate your cells, you can get away with using a pretty low concentration of antibody. So you don't always have to use a saturating concentration for phenotypic markers, as I'm sure you all know. So for instance, here I would say 130 counts per cell, as you see, for half a microgram per mil is probably perfectly sufficient to separate my 45.1s from my 45.2s. But it's really, really convenient to have very clean biological positive and negative control populations. And this becomes fairly important when you're trying to determine the background of your antibodies. As you all know, antibodies pretty much always have some level of background staining. And if you try to use your antibodies at too high of a concentration, that can become a problem for you. So looking at a newly labeled anti-CD3 antibody, comparing it to its expression on T cells or B cells, you can see that increasing the concentration, in this case 16-fold, doesn't make a huge difference on the CD3 labeling for the T cells, but what does start to happen is your B cells start to label with CD3, which is fairly inconvenient when you're trying to gate those populations very cleanly, and especially when you're trying to use some of the unsupervised methods of data analysis that Sean briefly touched on at the end of his talk. So using antibodies at high concentrations can be equally problematic as using antibodies at too low of a concentration. Again, titrating is important. And then, again, just to illustrate that a lot of times a low concentration will really provide you the same amount of information that a high concentration will. You'll see here CD44, which is often used as a memory marker in T cells to separate naive from effector T cells, will give you sort of a trimodal distribution on primary T cells, some really bright CD44s, and then some intermediates, and then some lows. And while that's not so evident at my lowest concentration here, it really is evident at every concentration higher than that. So again, you can save yourself a lot of antibody by here staining at 0.5 micrograms per mil instead of 8. So hopefully that was a little bit helpful to illustrate some of the parameters that we try to optimize on when we go about labeling and titrating our antibodies. So troubleshooting. What do you do when you've just labeled a new antibody that you're super excited about and it just doesn't stain that well? What I generally do for my first step is to check the staining on fluorescence. So I'll get a good secondary antibody against the host of whatever antibody I'm using and stain some cells with either purified or CyTOF labeled antibody throw on a secondary, run it on the flow cytometer, make sure the staining patterns look similar. If they don't look similar, if your CyTOF conjugated antibody loses all of its staining, then that probably means that the reduction either wasn't done properly or isn't going to work well for that specific clone. And there are some workarounds for that. If your signal does look really similar on a fluorescence-based flow cytometer, then what probably happened is you either forgot to add the metal, the chelation didn't happen properly. I would just go ahead and relabel that antibody because it really should work. So some alternative labeling strategies if you do come across a clone that you really want to label but just isn't going to work through this basic chemistry. The first is to use a biotin-streptavidin system. There's now a streptavidin. So streptavidin doesn't actually have cysteines, but Evan Newell from Mark Davis's lab has engineered one that does have cysteines. So you can use that to label using fairly similar chemistry to what I've shown you here. And the second, you can use an anti-fluorophore secondary system. So you can use your antibody of interest conjugated to a fluorophore. There are commercially available secondary antibodies against PE, APC, FITC, probably per-CP, I would guess, since it's also a protein. Maybe not. No, not per-CP, just PE, APC, and FITC. So there are 3 options for any given panel of antibodies that you don't actually have to primarily conjugate. You can just throw on a CyTOF conjugated secondary. Something that Sean touched on fairly briefly at the end of his talk, nanoparticle reagents. These are sort of still in development, but the ones that we can use right now off the shelf are Q-dot labeled antibodies. Q-dots are primarily cadmium-based nanocrystals. And cadmium has a molecular weight that we can detect on the mass cytometer. So here, as anyone who's done high parameter fluorescence experiments, and that seems to be quite a lot of you in the audience, you've probably played around with Q-dots at some point, because they do have fairly favorable emission spectra, tend to be pretty narrow peaks. And the larger the nanocrystal, the longer the wavelength that's emitted from a Q-dot. So in this experiment, what we did was to stain the same cells with 5 different commercially available Q-dots of different wavelengths. And as you can see, the redder you get, the larger the nanoparticle is, and the more metal we can see on the CyTOF. Now, it's also a little bit stickier. So again, we tend to use either 605 or 655 on the CyTOF rather than 800, which would give you the brightest signal, but is quite a bit stickier since it's bigger. So we can use Q-dot labeled antibodies, and hopefully more nanoparticle-based reagents are on their way. It's a really convenient way to boost signal. And then here, just showing you a bit of primary data, Q-dots are not isotopically enriched, as you can imagine. That's an expensive process, and for its fluorophore-like properties, it's not essential. So if you throw on a commercially available cadmium-based nanoparticle, you really see signal in a number of channels from 110 to 114, and we can sum those to get some sort of integrated signal across them. As I mentioned, we've also developed a viability stain here in the lab, and it's cisplatin. You've probably heard of cisplatin in the context of cancer chemotherapeutic, but it has the nice property that a lot of viability dyes do, that it'll translocate very quickly across a permeabilized membrane and very slowly across an intact membrane. So we can spike in some cisplatin for a short period of time and wash it out, selectively label all of our dead cells with the reagent, and cisplatin has a molecular weight of 195 AMU, which we can measure directly on the CyTOF. A couple other reagents that we commonly use, the DNA intercalators, as I mentioned. These are also now commercially available from DVS Sciences. There are 2 different ones, iridium or rhodium based. So we can label all cells with DNA. And then the barcode reagents we've developed here in the lab, you'll hear a bit more about tomorrow. But again, that's a way to pool samples and save yourself some time as well as some technical variability. One other reagent that I haven't added here, but I should have, that we've started playing around with in the last year. How many people have ever done BRDU labeling on fluorescence? Quite a number of people. So it's a really convenient way to measure cellular proliferation. BRDU analog of uridine gets incorporated into DNA. And you probably know that on fluorescence, you have to use an antibody against BRDU. Well, IDU has very similar chemistry, iodine deoxyuridine, and iodine has a molecular weight of 127, so we can actually measure IDU incorporation directly on the CyTOF. So that's kind of a nice workaround to do proliferation type assays by CyTOF, very comparably to how you would do it by fluorescence, actually a bit easier. Yeah, so those are some of the tools that you might think about using if you're designing CyTOF experiments, how to develop them, some considerations for how to optimize them. And I'd be happy to take any questions if anyone has them now, or of course, for the remainder of the course. Yes?
How would you titrate like a marker that doesn't have a clear positive and negative, like cytokine receptors? They just shift a little bit, so it's not like, two clear populations.
Yeah, it's tricky. It's tricky. If it's a commercially available antibody that you're fairly confident in, I would compare its distribution by fluorescence and mass and make sure that it looks very similar, and then I'd feel comfortable using it. If it's really a new reagent, you might think about something like an overexpression system or a knockdown to really validate it well. Or if you're working in mice, there are a lot of cytokine receptor knockout mice, so you could probably get some cells from one of the labs that's generated them. Mm-hmm.
Once you make your reagents, are they stable for a long time at 4 degrees, or do you store them in some other fashion, or do you know how long they're stable?
Yeah, so I would say every antibody I've labeled has been stable at 4 degrees for at least a year. I've had some that have been sitting in the fridge for like 3 years or longer that are still perfectly fine, and I've had some that after a year start to lose a little bit of their signal, but never had a problem a year or shorter at 4. Mm-hmm.
Say you have something which has a really dim signal in conventional fluorescent flow, something like CD25 or something. You pick your best metal, you validate that the antibody works, it's not being destroyed by the conjugation chemistry. Do you have any other recommendations for how to get the best signal out of signal?
It's something that we're trying to actively develop right now. I would say there's not a great workaround right now. You can use streptavidin, a biotinylated antibody in strep, which will give you a little bit of amplification, since most biotinylated antibodies have multiple biotins. But yeah, not a great solution right now. We're really hoping that the nanoparticle-based reagents will help us in that regard. But yeah, CD25 on mouse works pretty well. On human, it works, I'd say, decently well. But yeah, there are a number of things that we would like to be able to measure with greater dynamic range than we're able to right now. But that's generally the case on fluorescence-based flow, too. If you have to design your entire panel to have CD25 on PE, it's still a pain in the butt, All right. Well, thank you.